Welcome to this panel discussion that's going to follow up on session 2. My name is Dr. Peter Gottlieb. I'm a professor at the Barbara Davis Center at the University of Colorado Anschutz Medical Campus. We have a really great panel with us, so I'm going to let each of those members introduce themselves. I'll go around my screen. Matthias van Hereth, if you could.
I work in the Global Chief Medical Office of Novo Nordisk and also at the Diabetes Research Institute in Miami. And I'm very passionate about finding a prevention or cure for type 1 diabetes and certainly make the life for all the patients better. Thank you for having me here, Peter.
Thanks. Josh?
Senior Director of Research here at Breakthrough T1D.
To participate. My name is Laura Connect. I'm an endocrinologist. I'm also the Group Principal Clinical Research Director at Sanofi.
Lauren, if you could.
My name is Lauren Wood-Heichman. I'm a clinical reviewer in the FDA's Diabetes Division, part of the Diabetes, Lipid Disorders, and Obesity Division, Office of New Drugs. And I've been involved in the review of therapeutics being developed for stage 2 and 3 type 1 diabetes. And thank you so much for allowing me to be involved. I think this is very important discussion, and I'm excited to address gaps in care in type 1 diabetes.
Carmela.
Hey everybody, my name is Carmela Evans-Molina. I'm an adult endocrinologist and faculty member at the IU School of Medicine in Indianapolis. And I'm also an investigator in the type 1 diabetes TrialNet program. So I'm delighted to be here as well. Thanks.
I'm also very pleased to be here. I'm the CEO at Diamond Medical, a company founded on the mission to cure type 1 diabetes. So this is obviously a very important topic for us.
Thank you all for being here, and I'm looking forward to our discussion. We've just had 3 beautiful talks by Drs. Greenbaum, Bottolino, and Van Hereth. We want to expand on the points that they were raising during the session. At this stage of disease, maintaining C-peptide, keeping someone in a honeymoon state, as a number of us who are clinicians know, can make such a difference for our patients. And I think as we heard in session 1, from the people who have it, it certainly makes a difference too. So, really, I think what we would like to do is to try to expand a little bit on what we need to do. The TOMI study, which will be discussed in more detail tomorrow, was alluded to earlier in Matthias's talk, shows that if we do a large enough study, we can see C-peptide correlates to A1C. But as we're going to discuss, and as we know, that's so hard to do. I mean, those are very large numbers that were presented and certainly not something that's feasible for us to do currently. So how can we move forward? What can we do to make things work better? Carmel, I'll start with you if that's okay. Your work's focused on the beta cell and how to measure function, ER stress, as both potential cause and effect of autoimmunity. How would you characterize where we are as using C-peptide as an outcome measure? Let's start there.
Peter. So as you mentioned, we've been interested in the beta cell, and one of the areas of our research is to try to identify things that might be secreted from the beta cell that tell us about the health status of the beta cell. And we've looked at lots of novel biomarkers, but really when you get down to it, there's nothing that beats C-peptide, I think, in telling us about how the beta cell is functioning and its health status. So I think a lot of the talks have really highlighted what we do know, and that when you look in large enough data sets, that you can definitely see an association with preservation of C-peptide in very important measures like glycemic control, protection against hypoglycemia, glycemic variability. So I think that raises a very important question on how we might utilize these past data sets to inform future trials, and really what is the purpose of trials. And I think all of us here really would like to see something approved for stage 3 type 1 diabetes. One of the questions that I think that always comes up is, well, what about the safety? I think we do have really very compelling data to show that we have benefit. What about the safety? One of the hats that I wear in TrialNet is chair of the long-term follow-up study called LIFT. And we've had a lot of opportunity to interact with people who have participated in trials. And with Laura Jacobson, we've been looking at a combined analysis of our 14 trials that were performed in TrialNet and ITN. And really, if you look at that long-term data, it's very, very reassuring that people don't have long-term safety signals from past participation in trials. And certainly while we perform those trials, there's been nothing unexpected or in our type 1 diabetes population. So I think that there's a long track record of safety. This new data really, really highlighting benefits. So I think we're at an important point and a crossroad in the field to think about how we move forward.
The things that was brought out in the panel discussion in session 1 and indirectly through the talks as well was really that idea of benefit and risk and how we weigh that as we're moving forward on the different therapies and things. And I think, our experience, which now it sounds like you guys have collated, really supports that. I think the industry experience supports that as well. So I think, again, now it's this issue of trying to find the benefit. I don't know if any of our industry partners would want to comment there.
Yeah, I can comment on that. I mean, the space, the way we see it, is pretty well mapped. That the ideal therapy, if we're talking stage 3, right, will somewhere live between vitamin D, which you could argue is easy to give, right, and a non-myeloablative bone marrow transplant, which arguably you could argue it might not be applicable to any of the patients because of the risk-benefit balance. And I think because these trials are cumbersome and complicated and combinations at the end of the day might be needed, I think the wish is that if the risk is tolerable, let's say if there's an antigenic tolerance or if there's an induction therapy where you're not permanently staying on certain drugs, that we need to build avenues where we can collect data we don't have yet, and arguably we would want to collect, that we can collect some of these data post-approval. That would be very helpful. And that, of course, has to be balanced in terms of the risk of the intervention, as far as we know, after phase 1, phase 2, depending what drug it is. And it will, of course, vary from drug to drug. So there cannot be a blanket recommendation that that's how it is. But sort of the general wish is to have a very open thinking and discussion about it, just to provide a runway for stage 3 drugs. Because it's also my belief we can present tremendous benefit to patients, but we just need the runway and we need to collect the data then. And we would be wiser 10 years from now, but we just need to get the show on the road, so to speak. Right.
Yeah, well, I think the FDA has sort of had put a mandate to the field, and certainly I think both TrialNet and ITN have sort of tried to address that over the years with the studies that Carmela was mentioning. So I think finally getting to put some of that data out to support that, I think, is really important. And as we know, I think the FDA has asked Sanofi to continue to follow up on the Tzield patients, and that process is ongoing. So I think certainly as we build more and more of that database, we'll understand the risk aspect. I guess the thing, as we discuss, and it's been sort of the hang up, is the benefit. How do we define that benefit? A number of us on this call really do feel that C-peptide fits the bill. I'll just put out, since today couldn't join us from Europe with his talk, that the CGM metrics would certainly are improving, are starting to, I think, point to directions that can both be outcomes, but I think also probably would confirm the effects of C-peptide, for example. And the time and tight range, I think, from what I'm hearing, and I think we're hoping will come through, is potentially one of those. Carmela, what do you think? Is that something that's ready just yet? Is there data supporting time and tight range, or even time and range versus C-peptide, that you're aware of at this time?
Well, I think that's certainly an area where more data is being developed. And even within TrialNet, we only have a handful of studies now that are incorporating CGM, but we have lots of CGM data, I think, from many different sources that can be analyzed. I think about the CGM data maybe in the same way. You're talking about several different metrics, taking that data and looking at it in different ways. I think that we've also seen really interesting analyses when we take our standard C-peptide data from mixed meal tolerance test. Area under the curve gives us a kind of blunt measure of how the beta cell is functioning. But when we take more nuanced looks at how the beta cell is functioning using different modeling techniques, et cetera, we can see really more substantial changes in the glucose responsiveness of the beta cell. So I think in my mind, perhaps more nuanced look at the beta cell and how it's functioning and responding to challenge combined with CGM is really a good way for us to think about going forward. And then obviously mirroring that with metrics like hemoglobin A1C. I mean, I think we all know it's one of the challenges when we have a clinical trial population is that they're a highly motivated population, and their glycemic control is going to be probably a lot different than usual care. So it's very difficult in the time frame when there is already a honeymoon, when glycemia is much different than if you look 3 to 4 years after diagnosis, that you're going to see big differences in that measure. And I think we probably can do a lot better about just getting more of that longer term data as well.
Yeah, I think the point, today was making, which I think is probably correct, is that what we're looking for has to be subtle. The idea at this stage that you can find, at least in a short-term way, A1C differences, hypoglycemia is just not there. Our current technology is really helping to take care of that. Hopefully earlier and earlier diagnosis is helping to take care of that too. But so relatively, we do need these other measures. In Carla's talk, she was bringing up the idea of QR as sort of one way to actually, in some ways, use that large amount of data as a functional control, if you will. One of the questions raised by one of our comments through the course of that session from Jeremy Gale was that do you still need placebos? I don't know who wants to maybe jump at that. Josh, do you want to take a stab?
Whether or not we still need placebos.
And also your thoughts on QR.
Yes. Well, let's start with the placebo question. I think that type 1 diabetes is very fortunate to have large amounts of data from natural history studies, from clinical trials, from multiple sources that a lot of other indications don't have. So I would think that if at all possible, leaning into some sort of adaptive trial design up to and including synthetic placebos should be something we consider if we want to try to improve clinical efficiencies and get these therapies to patients as quickly as possible. With regard to QR, I think that one of the unique places we are right now is we have a number of trials that have been completed in the new-onset setting, and an ability to go back and clearly define what a responder is, who a responder is, and having a way to measure those across trials according to QR measurement based upon C-peptide is really going to help us move to the next generation of therapies for T1D, whether it be increasing durability, identifying likely responding populations, or leaning into more precise medicines.
Yeah, no, I think from our perspective of looking at this over a period of time, the QR I think holds that potential, and as we know, there's work that's being done by some of us here to actually address that, and hopefully that paper will come out later this year to try to support those efforts. Lauren, just maybe your initial sort of take on where we are at this point in terms of, the responsiveness of the agency to some of these sort of ideas and how you all are trying to evaluate risk benefit in this time.
Yeah, I think that I can discuss a little bit more of our response to some of the earlier speakers. There were a lot of really exciting points and clear resounding messages that I think we have alignment on. I'd really like to echo what other speakers discussed. There's a clear and substantial unmet need in the treatment of type 1 diabetes. And, I'm preaching to the crowd here, preaching to the choir here, but moreover, about 20% of children, 30% of adults meet glycemic targets in high-income countries. There's an estimated 16-year reduction in lifespan in those diagnosed younger than age 10. There are many other obvious reasons why there's substantial unmet need, including the high level of disease burden among patients with type 1 diabetes that we heard about, despite advanced insulin delivery systems. So I first want to reassure that we hear patients, physicians, and advocacy groups loud and clear. We understand that there's actually quite a tolerability for risks and uncertainties in this space based on this unmet need. And I agree with and take the opinion and lived experience of those patients with type 1 diabetes seriously. Also want to clarify that we do not think these recent advances in glucose management and insulin delivery tech really meaningfully changes the unmet need in this area. And our tolerance for risk and uncertainty. So that's kind of my piece on risk. And I agree with what you guys have discussed about tolerance for risk in this area, and for addressing the benefit through creative trial designs. I think that we have an openness to different ways to address the challenges in this area. But I do want to reflect something that previous speakers also touched on, this quagmire, the uncertainty of the magnitude of true benefit is kind of the main issue that is echoing in our minds when we're evaluating the benefit and risk assessment of a specific treatment. And so the real situation we don't want is if a clinical program demonstrates a specific magnitude of C-peptide preservation, but doesn't demonstrate a clinical benefit associated with that magnitude of preservation. It would be very challenging to justify a positive benefit risk assessment for a drug that also demonstrates meaningful risks, even if those risks are not ultimately seen long term. And so we understand there's limitations to evaluating these clinical endpoints in this population related to trial design. And so I think creative ways to address that are important. One of the things that I heard through the earlier speakers is that our patients really deserve to know what benefits are associated with treatment, just as much as they deserve to know the harms that are associated with treatment. So within this framework, this concept of clinical benefit, which we're trying to achieve, is that that concept of clinical benefit is how patients feel, functions, or survives. And while our published guidance lists A1C and reduction in number of ADA level 2 and level 3 hypoglycemia as acceptable endpoints, echoing all of the community here, we hear clearly that there are many clinically meaningful endpoints reflecting a reduced disease burden, and agree that these should be included in the benefit risk assessment. And we're open to considering other endpoints beyond glycemic control that could capture this reduced disease burden, like removing the need for or reducing the number of days where prandial insulin is required, or improvement in other certain aspects of daily life that might be captured in patient-reported outcome measures. And as Dr. Matthew mentioned, Chantal Matthew mentioned, there may also be other endpoints that better characterize this meaningfulness of a prolongation of the partial remission or honeymoon. And so we acknowledge scenarios where even a therapeutic may demonstrate equivalent A1C effects as standard of care therapy, but improve other important aspects of how a patient feels, functions, or survives, and express openness. So that's my 2 bits there. I would love to hear your thoughts on those potential solutions.
Oh, look, I think that the thing that I think characterizes, what's going on today versus maybe a number of years ago is how much I think consensus we all have. I think we're all looking for this way forward. I think what you've just talked about are the things we're all looking for too. Certainly, I think there's a number of us in the field that didn't focus as much on quality of life measures, patient-reported outcomes. I think we didn't think that would be significant enough. And certainly, I think now that's been added to a number of our trials to try to really help improve things and all the rest. So I think, what you're saying, I think makes a ton of sense. I think it's the kind of ways that we'll get there. Because again, I think all of us sort of have the knowledge, and I think that'll be brought out tomorrow in tomorrow's talks, that this does work. It takes so much time, or it takes so many numbers of patients. So what can we find in a timely way that can really support what we're talking about? I think one thing that we haven't totally the presentation, that obviously some people have very high C-peptide coming into it, and as was mentioned by Carla, that certainly makes a difference in terms of how we can measure where they're going to end up over the course of either 6 or 12 months in her work. But certainly, I think, whether you're a child or an adult, I think it's part of this, and is something that I think we have to weigh a little bit because of could we come to consensus on some subpopulations or some other groups as a way to start the process? I think since Matthias, you advanced that, let me throw that to you, and then I'll have a few of our other panelists comment on that.
Yeah. I think, I mean, you are start getting into biomarkers and slicing the pie up a bit. I mean, that, in my opinion, would already be quite an advance, because it might be, like we deal in this disease and other diseases, the more organ you have preserved, so to speak, the bigger the benefit is. And it pushes things ultimately into stage 2, and it's a testament to that the first drug we have approved, thanks through big heroic efforts of prevention in Sanofi, in that case, is in stage 2, right? And that's good. It would be, I think, that would be reasonable also based on the TOMI analysis, that higher C-peptide levels maybe one could consider, because they correlate very well with HbA1cs based on that analysis, that for sure could be a criteria one should consider for approval. And then the other argument is, and I'm sure we don't all agree on that, and it has, of course, many things to weigh if we're talking about post-conditional approval obligations to observe certain things. It, of course, depends what and how much and how long, right? But my inclination, my personal inclination, is that if it's too short, that also might not be good, because we might not learn things that only occur 5 years post-approval. And then, of course, you have to argue, well, how many things do we need to observe? And I'm sure we will have diverging opinions, and just because of the burden of doing that. But maybe that's, if we do go these routes, we can slowly get to a very good consensus and collect data. Maybe in some cases we really don't have at this point yet, but we will need for the future that then even open the field more, right?
Laura, Ulf, thoughts on this or other things that you wanted to discuss?
I can, obviously for us at Diamyd, it's been completely central to focus on a subgroup. So becoming more like a precision medicine thing because of our history. I mean, we were in phase 3 15 years ago, treated broadly in stage 3, and we saw a clear trend, no safety issues, but no significance. And since then we learned that it seems to be the MHC complex HLA genetics that's the key for us when we treat with an antigen. So we are basically now focusing on a genetic subgroup in an orphan designation. So we are really moving into rare disease space now. But that's really been our way forward. And also I think where we really found alignment with the FDA in our dialogue, since we had the orphan designation a long time ago, but now we go for the genetic subgroup with a clear scientific sort of rationale behind it and data that supports it, plus the safety profile. And that has led to the fast track designation that opened up the discussion about the potential accelerated approval process that we also have aligned around. So I think also when touching upon the regulatory dialogue, it's really been a journey for us, I think both ways, both for the FDA to understand us and for us to understand the FDA, and that we really, together with the field advancing, I think, like you also, Lauren, highlighted, that the FDA really understands the unmet medical need. And I think our fast track designation is partly that there is true alignment right now around type 1 as an important disease that needs new therapeutics. And we've been able then to really focus in on a responder group, which I think in general is like the way forward for any asset in a complex disease. Find the responders, don't treat unnecessarily individuals who don't benefit. If you find the ones that benefit, that's always the best thing. But that's really been key to us, and we obviously echo everyone else here feeling that C-peptide is the target. That really is the marker for showing that you sort of slow down or halt the destruction of a microorgan, which is significant. And the other measures are very challenging considering, again, clinical trial participants are highly motivated, treated at academic centers, treated to target. So it doesn't reflect the real world. So that's why really C-peptide should be the target. And then I agree with Matthias, maybe the long-term real-world follow-ups that will sort of show the long-term benefits, but we need to come there first. And then we're only scratching the surface currently on what I think is possible with all these different therapies that we are talking about. But we need to get approvals, start increasing the number of individuals that we are therapeutically treating, and then I think the field will advance very considerably.
Thanks, Flora.
Yeah, no, I agree with what's previously been said. You know, it is important to see who's going to respond, what different subgroups can potentially benefit from a therapeutic agent. But it's also important to bring therapeutic options to the patients, right? And to look at disease modification, and to make sure that you can have a clinical trial that can have a probability of success and can be feasible and enroll and retain people. And so it's such an exciting time for there to be so much interest in the type 1 diabetes space, but we want to make sure that we're able to bring therapeutic options to people.
Lauren, any comment from you in light of all of this?
Yeah, whenever you ask what developers could do to assist and inform these considerations in such a it's almost painful to ask a drug manufacturer to limit the population that may potentially derive the most benefit. But whenever we're having such a challenge in finding a clinical outcome measure that's for a treatment effect in this population, that's one of the first things that comes to mind, is limiting the population to those who could derive the most benefit, either specific HLA or immunophenotype, in order to limit the heterogeneity in the disease response, at least in this period where we're trying to find a placebo-controlled treatment effect of a drug. I do think that Dr. Van Herath made some good points about how, particularly for A1C, it might be something to look into, is baseline C-peptide that Dr. Greenbaum also mentioned, described in detail that a lot of heterogeneity is from baseline values. And so accounting for that by limiting population is one of the ways that you can account for that heterogeneity. But this is such a complicated issue, and I think coming together and discussing these challenges openly is really important. My other suggestion or thoughts that I mentioned briefly is to evaluate tools intended to measure patient burden really early on in the program, at an earlier stage of development than you think that they would be required, in order to evaluate their measurement properties early, so that if you have a patient-reported outcome measure designed to capture important concepts that you believe your therapeutic will demonstrate improvements in, you would want to evaluate those early and present them to the agency early. We have a whole division that's devoted to evaluating the ability of these to capture. So that's the other point. I'm sure there's lots of other different things that'll come up during discussion, though.
Thanks. Josh, I know we haven't touched. Matthias, come back. Go ahead.
Yeah, just a small comment. I mean, we have to. So your thoughts are extremely helpful, Lauren. First of all, this is very encouraging to hear. And there will be different determining variables. For example, this issue of MHC restriction is interesting because it's in a way binary, right? It makes sense with an antigen, and it makes sense also in terms of certain genetic risk. We will have variables that are low continuous, right? And then if they are continuous variables, you're going to go like, well, at some point we need to set a cutoff, and setting that cutoff properly, like a test, any clinical test you have, right? We actually need a lot of patient experience, and there's again the appeal that probably prior to approval or prior to conditional approval, one can probably have some idea, but not be super crisp on where that cutoff at the end might lie. And we will need that real world experience at the end of the day, in my opinion, to make the right decisions on that. That's something we should probably consider.
Carmela?
Yeah, maybe just to go back to the idea of heterogeneity, it's a tremendous challenge for our field. And while I love the ideas that we are, talking about precision medicine and subpopulations and targeting therapies, I think that is it's I think there are examples like Ulf was discussing of things that are more proximal, but I think a more immediate need, as I see, is that we work towards approval of our most promising agents that we have been testing for decades. And I think, if you think about what we know about the pathophysiology of type 1 diabetes, we know so much about the acute pathophysiology when someone develops type 1 diabetes. And I can't think of any other example where we watch the destruction of a microorgan so passively, and we don't really harness that chance to give something that we know has a benefit in preserving C-peptide. And our mismatch is not that we don't believe that we can preserve C-peptide. It's really that preservation of C-peptide in the 1 to 2 years after diagnosis and all the rich data that we have about the benefit of C-peptide in long-term disease. And so what we really have is a mismatch in our observation period of our trials and where we know that C-peptide makes the most benefit. So to me, it doesn't seem a big leap of faith that if you send a person into long-term type 1 diabetes with more C-peptide, they're going to do better. But it is challenging to follow people and to ask them to be engaged in a trial for 5 to 6 years so that we can prove that. So I think we just have to somehow get past this mismatch, but understand that we should treat type 1 diabetes as an endocrine emergency that needs to be intervened on. And the fact that we have 30, 35 years of experience with drugs that can halt that process to an extent, and we're not offering those to people, I think it's a problem that we have to move past.
Laura?
And building on that a little bit more, knowing the history, the rich medical literature that exists showing the benefits that exist with C-peptide preservation, and knowing that in new-onset T1D, there's still functional beta cells that are there to protect, and losing that time to allow that organ to allow those beta cells not to be protected while you're trying to slice and dice can be a little bit difficult. And so I think it's important to look at the fact that these are disease-modifying agents and the benefits that can come from that have been proven and that have been accepted in the medical literature.
Josh, your thoughts on this, but also I think on, we haven't really touched on other potential correlates of C-peptide, other immunologic sort of signals that we are developing.
Yeah, I think the immunological data in type 1 diabetes is very telling, right? We have a strong understanding, as Carmela said, of the biology of T1D, better than most autoimmune diseases. And we know that at its simplest point, the loss of the functional insulin-producing beta cells is the result of an autoimmune attack on these cells. There's been a number of studies that have highlighted a correlation between preserving C-peptide as a measure of continued beta cell function, and then specific immune changes or correlates that are related to the mechanism of the drug or pathophysiology being tested. You think of the work of people like Alice Long who've shown the exhausted CD8 phenotype, right? That really correlates with those in the teplizumab study that had stabilized C-peptide. We saw very similar results, Peter, as you're well aware, with Aleficet, that is similar exhaustion phenotype in these CD8 T cells. Recently, with the ustekinumab trial, we saw this inflammatory CD4 population, the 17.1s as we're calling them, is associated with the preservation of C-peptide in those patients. And consequentially, that population was also observed in the Aleficet study. So, I mean, there's many examples of this, but really, I think what this highlights is that the immune cell populations that are the intended targets or part of the pathways that are being targeted by these immune interventions are displaying strong correlations with the functional beta cell preservation as we measure by C-peptide. And I think really the takeaway from that is when we think about the kind of evidence that are necessary to help us establish surrogacy, I think that this data actually does support the biological plausibility of C-peptide as that marker.
Okay. Sorry. Oh, please.
Yeah, I just wanted to go back to what Carmela was saying, because that really resonates. I think also the whole, just having that picture in front of you that there is a microorgan that is being destroyed, like you have an irreversible autoimmune attack and you do nothing about it. We haven't done anything about it for 100 years, while we could do something about it. So that's, I mean, that needs to be changed. And then I also think I heard I read some text about, well, how it's been, because we are really in rare disease. I mean, when we look at stage 3 recent onset, it's an orphan designation, and in rare diseases, it's very common to talk about like any incremental benefits are welcome and should be. Like, it's a completely different kind of discussion when you talk about rare diseases. But I think we need to realize that stage 3 recent onset type 1 is a rare disease. And like we just talked, I mean, if we can even slow down the disease to some point, it has a huge benefit. If you think about that, there is an organ that is being destroyed in the background, and we are just artificially trying to supplement and sort of with technology just cover the symptoms. But I mean, the disease is there, and endocrinologists are basically just titering insulin today, whereas they could actually treat the actual disease. So I think that should happen.
Yeah, I mean, and again, just to take it one step back, because again, it's also where I think all of us really want to go. Look, you go into stage 2, they haven't lost that many beta cells. And as most of us would think and believe, there's very little difference between stage 2 and stage 3 other than timing and where you catch somebody. And as we've seen in people who've been in our natural history studies in all the different countries in the world, it's all the same. As they start to go down that pathway, that's the opportunity. And it seems like a number of our drugs could work. So again, it's that opportunity that we need to create to be able to move the field forward. I think, apropos of this, I'll sort of throw it out there a little. When you think about transplantation, and granted, at the time, the alternative was death for people. So when they started out, they were happy when they got a few months. And then they were able to get it a little bit longer, and obviously similar for cancer therapy and the rest. And in some ways, I think, we're really in some of that. We know that we can do things, we can get an effect. It isn't lasting as long as we want, certainly a year or two at present, but certainly as was shown in some of the slides earlier today, it is a benefit. And I think the idea to build on that and to make it even better is really where we want to go. But you need to have the first steps to be able to get to the second steps. Matthias, go ahead.
Yeah, maybe a little plug, because of course you could argue that this is a disease where you have a beta cell dysfunction and an immune dysfunction, maybe even an attractor hypothesis where dysfunctional beta cells attract the immune system. That means at the end, we might need combination therapies. You could argue, how do you go about this? And we have been there, of course, and these trials are not easy to enroll. But if you have a combo trial, you need more patients, and especially in stage 2, even more patients. One way this could sort itself out, which would be beneficial for everybody, is if there are more drugs ultimately approved in the stage 2 and stage 3 space, then it becomes easier to rationally combine it. And I'm not talking about all of a sudden packing 8 immunosuppressants on a single patient. That's obviously not the point. But if there's different MOAs that might at least be additive, or in the best case be synergistic, that could be very helpful. And it will be helpful at the end if it doesn't have to all be done in an a priori combination trial. Safety, yes, but fully powered combination trials and then proving this over many years will just become, the bar will become too high. So that's one thought that might be very helpful for the field, because I strongly believe there's these 2 components to the disease. And rather than arguing, oh, this is going to even further complicate the whole matter, you could say, well, if you target one component and then others target another component and these things are approved, then it becomes easier to put this together. Anyways, just a thought.
And certainly there's some data coming this year that may support your idea, depending on what happens with verapamil, et cetera. So we'll all have to see how that plays out. Other thoughts from people on the panel, things that we haven't touched on that you wanted to bring up? Josh, from a foundation point of view, what do you think the role is right now for where you and Breakthrough T1D and other foundations can sort of help in the process? Oh, you're on mute. You're on mute, Josh.
They warned me not to mute. Obviously, clinical trial design is something we think about a lot, right? And putting in the context of how we approach it, we're first and foremost a patient advocacy organization. We just advocate in a large part by funding everything from early clinical trials through, or I'm sorry, early research through clinical trials, with the ultimate goal of bringing cures to the people that we represent. As we talk about this amongst ourselves, other foundations in the space, I think there's a lot of excitement that there's a growing number of disease-modifying therapies that show an ability to preserve beta cell function just after diagnosis. I think, though, that we're also acutely aware that there's a lack of substantial progress for these therapies beyond phase 2 clinical trials and moving toward regulatory approval and actually benefiting the people that are in need. When we consider funding a clinical trial, whether it's a novel drug coming out of an academic laboratory or working with an industry partner that might have a drug with established safety and efficacy in another indication or might have approval in another indication, we always run into the same question. And that question is, what is the viable regulatory and advancement prospect for this therapy? In order for us to allocate funding, and our funds are entirely made up of donations from our supporters in the T1D community. We need to make sure that the trial being considered can be run efficiently, and to have confidence in the trial design that it'll live up to the regulatory expectations. And I say this because it's also the same thing we hear from industry members that are looking from the space. So it tells that there's a common issue here for us to overcome. I think from our standpoint, the current status quo is an impediment to us realizing our mission to address the needs of all those living with type 1 diabetes or at risk. And our goal then is to make therapies that meet a high bar of safety and efficacy available to the patients who need them as quickly as possible. So what can we do to do that? We firmly believe that our role is promoting a reliance on science and taking the science and the data available and producing that science and data that needs to be produced to develop a regulatory pathway that accelerates the development of therapies and ultimately benefit the patients. That's our goal, and that's where we see our role being. We also feel that the overwhelming data available at this point points to C-peptide as a viable endpoint for the assessment of disease-modifying therapies in new-onset trials. And certainly we want to work with our industry partners, the clinicians, and the regulators to do what we can to bring that over the finish line.
Okay, thank you. Thank you. I think we just have a minute or two, at least in our formal time, although I think we can run over a little bit if we need to. I think one thing that was brought up in the chat, I guess, is that I'm just looking at some of the questions that came from Simi Ahmed. Is that the conditional approval of aducanumab in Alzheimer's, whether or not that represents a similar sort of situation to what we're talking about here for C-peptide. I don't know if Lauren, if you're familiar, you want to comment on that or just comment on the last few subjects we've been touching on.
I think I've been hearing a stance around the pathway that we outlined in the last C-peptide workshop, which accelerated approval, whereby which appears to be the primary pathway available. I think that whenever you relate it back to another disease state, with the accelerated approval pathway is designed to help us really understand those clinical outcomes that may not be achievable in a feasible trial design and try to accelerate access in an area where there's unmet need and there's a reasonably likely surrogate endpoint. We think this is a viable pathway, but I don't know if I can comment specifically on that disease state. However, it is a potential pathway. The other thing I wanted to weigh in on is with these conversations where you have an approval based on a reasonably likely surrogate endpoint, as C-peptide is an example, the magnitude of benefit is, and quantifying that magnitude that your treatment allows with a clinical outcome is really important, because you might have a analysis of all of the trials conducted, and you might see a general trend in a improvement in an endpoint, but for the specific disease, for the specific drug product, you're looking really at, does the magnitude of C-peptide preservation result in impact how a patient feels, functions, or survives? In a case where maybe reduces your insulin amount by 2 to 5 units a day, it might be not something that would impact a patient's feel, functions, or survives. Although I know that there's a lot of discussion, there's a lot of many different clinically meaningful endpoints. So I think that's the nuance that we're kind of discussing that influences those decisions on whether or not an approval is acceptable as for a specific drug. So I hope that addresses the concern a little bit without actually weighing in on that drug product.
You're trying to thread the needle on all of this, and we thank you so much for your participation today. Ulf, let me let you comment as well.
Well, I would say that in type 1 diabetes, we have a much better biomarker than in Alzheimer's, like the amyloid hypothesis, just a hypothesis, but C-peptide is the true measure of a function. So I would say we should be in a much better space than in the Alzheimer's, but there you have had the accelerated approval. Obviously, it's been a risk-benefit, I think, discussion in that field, which we can talk about as well. But on the other hand, also, I would agree with Lauren. In the end, it's the asset-by-asset basis and the totality of the data, both from the whole field, but also for that specific therapy that should be evaluated. And like Matthias also alluded to, there are, like you have an autoimmune component and you have a beta cell function component. So you might have different flavors of C-peptide preservation. So it's not necessarily that the same C-peptide preservation results in exactly the same benefits for depending on which therapy you use. So in the end, it should be a therapy by therapy basis, but we should have some kind of common guidance for the industry so that we can move forward. And I think we are quite aligned here. So.
And I think we're moving in that direction, which I think is a positive thing. I think there are some other things we haven't touched on. I think certainly as we talked about populations, the potential differences between adult and children that we really didn't get to too much in this discussion, but certainly is something where we do need to learn so much more about the diabetes that does occur in adults. And as we heard from Chantal, is now actually a large percentage of the people with the disease. But I think we're out of time, and I'd like to thank everyone for a really wonderful, very informative discussion. Speakers who presented their work, and our panelists, and also the people who sent in those questions. So appreciate it very much. I think, as I said earlier, I think what characterizes us all, and this is a nice combination of academia and industry regulatory agencies, is that we're all really trying to work together to improve the care of people with type 1. And we hope that this workshop will help us to move our field forward towards the new therapies to treat and ultimately prevent type 1 diabetes. So the workshop will continue with the wrap up, and thank you again for all your time and attention.
Thank you, Peter.